Hello everyone, welcome to the afternoon session of this IMI Open Info Day. If I could ask you to please take your seat so we can get started. So first of all, I would like to welcome all of you. It's nice to see so many people in the room interested in this interesting topic. I would just like to tell you first what's going to happen, because it's quite a busy afternoon. We will be focusing in this workshop on the translational approaches to disease-modifying therapy for type 1 diabetes. And first we will have a presentation from the representative of the industrial consortium from the FPR side. Followed by, of course, question and answer session. And finally, in the end, we will have the so-called elevator pitch session. We have 23 participants signed up for this elevator pitch, and this will be an opportunity for people who are interested in participating to this project, who have something to offer, who would like to join consortia or advertise their consortia when they need new partners, will have 90 seconds to present what they have to offer. So without further ado, I would like to introduce Dr. Anke Schulte from Sanofi. She has studied in Germany and Wolfgang Goethe University as well as Darmstadt. She has done her postdoctorate studies in University of Georgetown in Washington, and then she joined Sanofi in 2001, where she established a molecular pathology group within the functional genomic department. And then since 2010, she's working with the metabolic diabetes department, and he is the head of ISLET biology department there. So please, Anke, if you can tell us all some more details about this exciting topic.
It was a very kind introduction. Yes, it is me a big pleasure today to lay out to you the topic text we have written together with the FPR people who participate in the type 1 diabetes call. And we'll give you a presentation today which will give you the highlights and the key points which we think should be addressed in this call. So if I understand this one then. Okay, so let's start with a general introduction to type 1 diabetes, which to most of you will be already very well known. That's why you're here in this room. Okay, as we know, it's a chronic disease affecting worldwide more than 17 million people. And what we also know is that the incidence rate of type 1 diabetes has a very high flexibility between the different countries. Overall, you can say 40 out of 100,000 in Europe. But there are major differences, depends on the area of Europe you're looking at. And we also know that the incidence rate in the United States is about 25% lower, which shows to us already that the disease is very heterogeneous and the incidence rate based on the incidence variations. We also know that the increase in type 1 diabetes has tremendously increased over the last years, in particular in the people who are under the years of 5. The question is, where does it, so as on the right side you see a graph which shows you this, that the incidence for the children under the year of 5, is this a pointer here? No. Where's the pointer? Which is the pointer? This was the pointer. Has overall increased by 5.4%. This shows us that something has happened which lead us to the situation that the incidence rate has increased. And there are a lot of different factors involved, a lot of discussions about the environmental influence on this kind of increase, explaining this increase. Okay, how do we actually diagnose type 1 diabetes? In the beginning, a patient comes to the doctor and you recognize that the person has hyperglycemia as a result of the functional beta cell loss. At this stage, the patient has already lost a lot of functional beta cells. To distinguish type 1 diabetes from type 2 diabetes, the next step what we're doing is we're using our biomarkers, which are the islet autoantibodies. But we also realize that when we detect the islet autoantibodies, the patient is already on the way to develop type 1 diabetes prior to the detection. So based on the detection of islet cell autoantibodies, we are considering the disease an autoimmune disease. We also know that the disease is very heterogeneous. A lot of data has been accomplished over the last years when people had the opportunity to look into the pancreata from type 1 diabetes patients and realizing that the amount of beta cells which are actually lost in the patients can be very different from patient to patient, independent that they are considered as have a full-blown autoimmune disease. So there's a heterogeneity in the beta cell status as well as a heterogeneity in the immune status. What causes the disease is actually unknown, as we all know, and because of this, it's also not preventable at the current time. We know the disease is driven by various different factors, which can be genetic factors or environmental factors, which induce the existence of the disease. We know, based on the excellent studies which have been done over the last years, that there are around 40 loci associated with the risk of developing type 1 diabetes. We know that siblings of children who have type 1 diabetes have a 15-fold higher risk of developing the disease than from people who have no siblings in the family who have already developed the disease. We also know that 90% of the people who are diagnosed with type 1 diabetes have no siblings who have already developed the disease. Okay. So how do we currently deal with the disease? We have a very nice pharmacotherapy which treats the patients good, which is lifelong treatment with insulin. But we also know that this is a major obstacle to the patients. The patient has to go on average 150,000 injections through the entire patient life. And we also know that insulin therapy has a risk of inducing hypoglycemia. And whether you do not take this sharp enough, you clearly run into vascular complications. So we do have a therapy, but we also know the therapy has a lot of gaps and obstacles we are facing. There are currently alternative options. You can go by organ or islet transplantation, but you all in the room know also that this has a lot of limitations in the usability based on organ availability and depends on the situation that the patient has to go through immunosuppression. So what we do need, we need a therapy which allows us to really modify the progression of the disease in an optimal setting to cure the disease, which means we need a therapy which would allow us to restore the beta cells within the pancreas of the patients by a setting where the body itself does not attack via those kind of beta cells via the immune system. Okay, so I think here we have completely common ground. Okay. There's a lot of knowledge increase been happening over the last years. So we know we have excellent ideas how to tackle type 1 diabetes. There are different screws you can screw on. You could say you reduce the triggering of environmental factors. And we know there has been a lot of study been done on the cow milk effect on the development of type 1 diabetes and the influence of breastfeeding and how long you're doing breastfeeding with your baby. We know the idea is we could, in general, suppress the immune attack, antigen-specific as well as antigen-unspecific. We know we would like to have something which generates more beta cells, rescues beta cells, and even do a beta cell replacement. These are all very, very good points, and we all would like to address them. But we also have to make at this current stage a reality check. So the reality check at the current stage is that we have not been very successful to have a limiting, lasting success in prevention to progression of type 1 diabetes. A lot of excellent trials have been done, but we see the effect is not lasting. So this shows us that we have certainly a gap in the understanding of the at-risk patients, the patient who has already developed the disease, and missing in understanding in the difference between the patients who are responding in the trials and the patients who are not responding in the trials. So we do need to characterize those thus better. So we think we need to obtain more knowledge to understand the at-risk patients, to understand the heterogeneity of the disease, to define scientific-based new criteria in setting the understanding of the entry point of a clinical trial as well as new readouts in the end of a clinical trial. And we do have to admit that, as independent that there are very nice models available which we all have used to understand the type 1 diabetic disease in a preclinical setting, we also have to understand that we have to get a more realistic understanding of what a preclinical model can really tell us in translation to the human. Okay, so you can ask the question, why do we need type 1 diabetes call in IMI 2 to tackle this kind of points? We do think, in FPR people, we do think we can only solve this problem when we get all our brains together. We think IMI 2 gives us an excellent opportunity, and this was very nice plotted out already in the morning sessions, where you can really bring the people, the experts from different disciplines together to get this moving in the right direction. That's why the FPR people are committed and are committed to set this call, to work with all the different experts together to really change something here. So the lacks are, we have definitely a lack in lasting type 1 diabetes disease modifying therapeutics. We all face that. Patients face this. We do have limited insight in the disease triggering events, which lead then ultimately afterwards to the destruction of the beta cell functionality. But what triggers it first, prior to detecting the autoantibodies? What is the initial event? We have a limited understanding of the patient's heterogeneity and the disease status. And we need to have clinical trials set up which gives us a more comparable setting with the results of clinical trials because we have more standardized criteria. So tackling this kind of points, we think by doing this together in an IMI 2 setting, we can make solid progress in the understanding of the complex pathophysiology of the disease, with the ultimate goal then to be able to perform new mono and combination therapies in more optimized clinical trial settings. Okay, so on this slide, I show you the overall project description, what needs to be addressed when you want to apply as a consortium to this call. So one goal is that we definitely have to build a cross-functional clinical and research initiative together. We want to bring the clinical people together with the research people, more together as we already are, to gain deeper insight to the heterogeneous phenotypic characteristics of people either at risk of developing the disease or have already manifested the disease. This, we want to map the interactions between the immune system and the pancreatic beta cells. We need to understand them both together better. And we certainly need to focus on the environmental changes having an influence and potentially responsible for the increasing incidence rate for type 1 diabetes. We want to build a European clinical trial network, and in this network we want to test novel approaches and standardized settings. Okay, so let me guide you through, we call it now the 3 objectives, to give you a little bit of a structure how we think this very huge endeavor should be addressed. Objective 1 would be to start from human material of people who have already at risk, who have already developed or live already with the disease for a longer time, collecting those samples retro as well as prospective for about 3 years. Certainly this only goes if the cohorts which are already available to all of you would be used and follow up in a prospective setting. Based on those samples, we propose to do really in-depth omics analysis of this material. But omics analysis is one point, so meaning proteomics, metabolomics, phosphoproteomics, transcriptomics, this is lipidomics. This is one point, but we also need in parallel the in-depth phenotypic characteristics of those patients at the different stages of the progression of the disease or at the onset of the disease. So, bringing all the health records from those patients together, as well as all the functional tests, who looks at the immune status as well as at the beta cell status. So this will be a lot of data. So this will only work if we have a systemic large database repository built who allows cross-functional data mining. And this was also very nice pointed out in the beginning in the morning, in particular for the type 1 diabetes call. It is very important also to envision imaging tools, which would allow us better to really control and visualize the beta cell status in the patients. So moving forward on the imaging side is a very important point also in this topic, as well as to create novel diagnostics and potentially also better devices to treat the patients. One other objective of the call is the situation that we do need to have a better understanding of the translational model we are using, reflective for the human disease. In particular, as you see here, it's not written preclinical animal type 1 diabetes models, it's the preclinical models. Can be animals, can be humans, human cells. Objective 3 is, if you collect all this data, as a result of those analysis of those comprehensive data, the goal would be then that we are able to set new trials, testing new mono and combination therapies for the positive effect in the patients, based on standardized criteria in those trials, having hopefully developed new starting points as well as endpoint criteria for the assessment. In the result of this trial, samples again should be collected to see whether we find biomarkers to better distinguish between the responders and the non-responders. So we do know that this is a very complex task we are laying out here. Okay, so the key deliverables of the full project are improved understanding of the heterogeneous disease of type 1 diabetes in their immunological and beta cell biology underlying pathophysiology. Integration of complex clinical and standardized molecular real-world data from at-risk and type 1 diabetes patients with support identification of new disease-relevant entry and endpoint readouts. We do want to have delivered after this end of the project a better understanding and the translational value of the preclinical models. The goal is to have a European clinical trial translational research network established, and within this network, new mono and combination treatments will be tested in the frame of the project. Okay, so what are the people, what is the envisioned consortium for us? What kind of consortium we are seeing here as FPR to be involved in all this very broad but however also very detailed described topic? So it will be a consortium of clinical and research departments, basic, translational, autoimmunity and beta cell biology focused people. There will be from the pharmaceutical side people involved who do very, very broad knowledge in drug discovery. There will be medical staff people. Small enterprises should be a part of it. Hands-on database specialists and data management people must be a part of this project. Without that, we will never get it to a point that we really get the essence out of all the collected data. Patient advocacy groups are absolutely one of the key points they should be applying and which should be a part of this topic to ensure that what we have done really reflects the need of the patient and that they are also informed of the developments which we are doing in this project, as well as experts in regulatory science and health technology and the European health authorities. So it's a very interesting mix of people which we are very much looking forward to have together on the table. Okay, what is the recommended modus operandi? How should we work all together? I think by doing this, we should learn from the very successful projects which we have already ongoing or completed in the IMI 1 setting. So it's highly recommended that the experts get organize themselves in cross-functional teams, which are organized in a way that there are work packages responsible for particular tasks to be completed, represented with people from the industry as well as the clinic and the academics. It should be all the time ensured that there are inter-work package knowledge transfer by the regular meetings which are held. And I think a lot of you will be already involved in IMI projects, and I can only talk for myself. I'm one of the key people in the IMI media project. This kind of interaction, if you have regular meetings, goes very well. And it can be only successful if there's an open communication between the people to ensure that no knowledge gets lost. And I think this also was very nice explained in the morning session, what the idea of e-media, of IMI is, how we should work together. Okay, so as I said, it is a very complex topic. And so what you find in the topic text is a suggestion from the FPR people, how we would think would be a nice way of organizing it. But please, it is a suggestion. If there's a better way, if you defined a better way how the project should be structured, then please feel free to make this recommendation. We made the suggestion just to give you an idea and to help a little bit with the consortium building. So in our way, how we see it is that we think we should have it organized in 5 different work packages. One is certainly only responsible, I'm sorry. For management, then we should have one work package which starts with the first part I have told you, with allowing in-depth phenotypic and omics analysis, testing of imaging technologies, setting up new devices, to really bring the information first to be able afterwards to make a new standardized clinical trial. So this would be one work package. Working with all these materials, doing all the in-depth omics analysis and the immunophenotyping, we thought it would be good to do this in a separate work package. Here, the samples should be collected absolutely standardized, and here are people who are experts in analyzing those samples. The result of this, everything what has been analyzed here, everything what has been collected here, must go in a data repository. So a repository must be built to allow the complex correlation analysis between the phenotypic and the molecular data, which could be another work package. And the result of all this would then feed in the work package 3, which would then lay out the plot to set up new trials, to test new mono and combination therapies under standardized setting. Okay, the length of the project, most of you will know, we are talking about a 7-year project. Why 7 years? Because we do think you would need at least 7 years to address all these points. And because I told you, we want to collect data first, doing the complex analysis, and then feed this knowledge to create new clinical trials. Therefore, the work packages will have different starting points. And the data repository should continuously go over the whole time frame to do all the comprehensive analysis. Okay. So, who are the FPR companies who set up the plot for this? Sanofi is the coordinator, and the co-coordinator is the JDRF. Together with the Helmsley Charitable Trust, GlaxoSmithKline, Novo Nordisk, and Eli Lilly. So these are the FPR participants and the associated partners who put the topic text together. Altogether, we are talking about a budget of 35,260,000 euro. It's equally shared by the FPR participants and by the IMI, which means for the applying consortia, we're talking about a budget of 17,630,000 euro to be given to the beneficiaries of the selected consortia. Okay, so let me come to one of my last slides. So where do we stand? The call which I have presented to you here today has been launched on the internet on the 9th of July 2014. Since then, you could read to this, I think it's a 10-page document, which describes in detail what we are aiming to do. The submission of the expression of interest from the different academic consortia, which will be formed, is the 12th of November 2014. Then, and this was also beautifully explained today in the morning sessions, independent reviewers will review the expression of interest. And a decision on the winning consortia will be taking place at the end of February 2015. The winning consortia get then together with the FPR participants to write the full project proposal. This has to be completed until the end of May 2015. Then the project proposal gets reviewed by IMI, joint undertaking, and the decision gets been taken whether this will be a project or whether this will be not a project to be started. But we heard in the morning, the success rate is close to 100. The gap to take, or the jump to take, is to have an expression of interest which will be the selected one in February 2015. Okay, if there are questions to this call, I'm happy to answer every question here. But if you do need assistance in writing the AOEs and getting together, please don't contact me. Don't contact the other FPR participants in this call. You contact the IMI office. As was also very nice explained prior to the lunch break. There are a lot of people who help you with building the consortia and help you with writing it all together. Okay, at this point I come to the end of my talk, and I hope it was helpful, and I'm happy to answer every question. Thank you very much.
Thank you very much, Anke. So now we have time for some questions, and please keep in mind that this is your very unique opportunity right now to ask questions from Anke, who is representing the team of the pharmaceutical industries, to get a better idea of what they are expecting from the applicants, what could be the tips to make your application stand out, or what are the requirements and things that you could include. So please, now is the time to ask questions. Yes?
Pieplers, Brussels Free University. It's very good to see that industry and big industry joins the goals of JDRF. And as you know, JDRF has been providing funding over a period of 30 years for goals that are listed in your slide. And many teams have been fortunate to receive funds. So it is a bit, the question comes up then that many of those goals have not yet received answers. Now you list those goals and you expect one consortium to provide answers to those basic questions within a period of 2 to 3 years in order to undertake innovative clinical trials. I doubt that that will be possible. On the other hand, I fully agree that novel trials are needed. It's very frustrating that we're able to pick up high-risk individuals from the population, and we cannot offer pilot studies or clinical trials. So I have an ambiguous feeling about it. On one hand, it's very good that the initiative is there, but I think that the approach is going to be too broad to have the questions addressed in by one consortium, and that when you're waiting for the omic data to start clinical trials, that will be practically difficult, I think.
Okay. Yeah, thank you very much for this good question. I agree with you. JDRF is the institution in the United States who drive and supports the type 1 diabetes research very efficiently. What we are hoping for, and that's also why JDRF is here the co-coordinator in this, what we are hoping for is that we bring the people more together. As a JDRF setting, it's very often single academic grants given to particular PIs. So the idea is, and this is our hope, that when we bring the different PIs together at one table, that we get better, that we are faster in getting it done. This is one very important point in this topic. That's also why JDRF was very happy to join this. I agree, we have very eager aims here. But it's not that we are, and the only thing, it can only fly if we are looking at already existing cohorts and characterizations which have been already done in a lot of the different PIs. We have to bring the information together. It's not so much that we have to bring we have to bring also new data, but also we have to bring the already existing data better together to allow their analysis. We do think that this will speed up the process. I hope I can convince you.
Thank you very much for this question. There's another one, two coming.
Roque Cardona from Barcelona, Spain. According to your presentation and due to the impact that type 1 diabetes has on pediatric population, do you consider that any proposal for this call should have an approach to pediatric population?
Yes, absolutely. In the first work package which I described, in which we think we do need to collect more samples and do better phenotypic characteristics, we're talking about people who are at risk, which include certainly young children. So,
thank you.
Please?
I'm interested in the pharmaceutical partners that have taken part. Some come with a much more metabolic portfolio. One or two have an immunological portfolio. I don't know whether you want to give us any, or able to give us any insight into their interests, in the sense that developing new immunological agents is extremely expensive and is unlikely to be something that can be developed in this timescale. But they are bringing new agents. Is there interest in exploring some of their portfolios, or that's not relevant to this call?
Yeah, I'm happy to. Very good question. So in regard to Sanofi, you may have also considered rather a company which is focusing on the type 2 diabetic aspects. In the situation here in this call, as you may know, Sanofi is together with Genzyme. And Genzyme is a very strong company when it comes to immune-mediated diseases. So I cannot give you specifics, but I can tell you that different companies, Sanofi as well as others, have already assets they want to test in 4 years in those trials. Does this answer your question?
Well, and my question is, should that be taken into account by the consortium?
Yes, you should, yes. So the information is, yes, there are assets which we think are ready to be tested in better standardized clinical trials in about 3 to 4 years.
If I can just extend that question for a moment.
Sorry.
I don't want to take too much time, but
no.
One of the issues very much with those sort of agents is the risk profile. And that's separate from some of the things that you've mentioned here. It's obviously to do with maybe Objective 3 and the clinical trials network, but really understanding things like opportunistic virus seroconversion rates, risk of PML, you know, how far can you perturb an immune system when in type 1 diabetes the bar is set very, very high in terms of no risk. So I don't know whether those elements of the consortium are going to be important, which are more, if you like, immunological risk and microbiological risk.
But the understanding is, if I'm saying that there are companies in the participants who have already something they are planning to test in 4 years, means that their tox studies on those have been ongoing or have been already done. Okay? Does this answer? Yeah.
Thank you.
You're welcome.
Another question?
Please.
I want to come back to your answer.
Yes?
So are you saying that that industry is at a stage where at a time where different companies, those that are listed in the consortium and others, have potentially interesting immune modulating or suppressive agents, that they are willing that their agent is tested in a comparative trial so that one company provides product 1 and the other product 2, just to see which one is more effective? Or the companies, have they agreed on that?
Yes.
Are there other questions?
Okay. So, I'm concluding from this that the message got through and it's all clear? Good. Thank you.
Thank you very much, Anke. So what we can do right now, since there is no other questions, if you still come up with some questions, we can hold them till the end of the session. We can move to the elevator pitches right now, and when we are finished with those, if there is still time remaining, we can take any other questions. Just to remind you that, of course, if you have other questions at some later point, you can contact us, like Anke mentioned, at InfoDesk, and you can also contact me directly, since I will be the scientific officer at IMI taking care of this topic from the IMI point of view. So please don't hesitate to contact us if you have questions or problems. So now, if the elevator pitch people are ready, I will be moderating this session, and I will warn you that I will be very tough on time, because we have to give everybody the opportunity to speak, and everybody should have the equal opportunity to speak, which means 90 seconds each. So I will call your name, you will receive the microphone, and as soon as you start speaking, this time will start. When your time is up, we will allow you to finish your sentence rapidly, and then we will go to the next person.
Tough lady.
So the first person is Rosel Arnale. Please stand up and start with your elevator pitch.
Hello, I'm Rosel Arnale from Barcelona, Spain, a children's hospital in Barcelona. And okay, the time is clear. And I will have enough time because, fortunately, I came with the coordinator of the diabetes program in our pediatric hospital, and he will do the speech later on. Thank you.
Very good, thank you. I will call the next person. It's Roque Cardona Hernandez. Very good.
Good afternoon, I'm Roque Cardona Hernandez. I'm the Pediatric Program Coordinator at Sant Joan de Déu Children's Hospital in Barcelona. Our institution is the largest pediatric diabetes center in Spain. We follow up more than 600 children with type 1 diabetes and about 70 to 80 new cases per year. It's an institution affiliated to University of Barcelona, and our clinical staff has broad experience in clinical trials involving new insulins, new devices, and new immune therapies. Our specific interest for this topic would be by recruiting patients and following up these pediatric patients, and by collecting and storaging blood samples. We have the support of the San Juan de Dios Research Foundation, who has a European accredited biological tissue bank, and we have also extended databases about our patients. So we would be very happy to join any consortium interested in pediatric population and to contribute to move forward to diabetes research. Thank you.
Thank you very much. Thank you for being perfect on time. Next person is Kristin Chichester. I apologize if I'm not pronouncing the names correctly.
Okay, it's Chichester. Hi, my name is Kristin Chichester. I'm from the Swiss Institute of Bioinformatics, and I've been working on the OpenFacts project, which is an IMI-1 project, which has built a semantic web data integration platform. We have information on proteins, diseases, pathways, small molecules, 3 billion facts, 14 million entities, 100 million mappings to about over 50 databases. So clearly the problem we solve is data integration. The importance of data integration is sort of emphasized by funding agencies that have started to think about requiring 5 to 10% of any grant to be spent on data integration. Now, you could just put up a database on the internet and then hope that that's the data integration solution for you, but you don't get the broad overview of the whole knowledge landscape. But if you do it with OpenFacts, oh shit, we have also already the established FPR groups and academic users. So basically an unfair advantage we have expertise in data integration, we have buy-in from FPR, and we know how to curate the literature, so we can add the appropriate semantics and ontologies to your data from the literature. And I think we're a good choice to have it on any consortium. Oh, I made it.
Great, thank you very much. Next person is Francesco Epifano. Perhaps missing. So we will move to the next person. It's Manuel Gia, or Gia. No. How about Fabio Grassi? It's coming.
Hello, I'm Fabio Grassi from the Institute for Research in Biomedicine in Bellinzona, Switzerland. I'm an immunologist. I'm interested in signaling by adenosine triphosphate in adaptive immunity. And in particular, so adenosine triphosphate, apart from being the energy currency of the cell, is also an extracellular molecule with signal. And we studied the role of ATP in a T-cell subset that is called T follicular helper cells, and it's very important in regulating this subset, and in particular it regulates a subset of T-cells which have diabetogenic potential, and so which accelerate and make the type 1 diabetes more aggressive in not mice. And also, so basically, and also in beta cells, ATP is very important for insulin secretion and insulin regeneration, and so we are addressing signaling by extracellular ATP, and in particular through P2X receptor, essentially in T cells, so in this subset of T cells, and in a subset which is characterized by CCR9, which is a chemokine receptor, and so we believe that addressing this signaling pathway is very important in type 1 diabetes pathogenesis. Thank you. It's very difficult.
Thank you very much. Thank you. Next in line is Matthew Groves, or Groves. No. Many people missing. Perhaps they were too nervous to actually go through with it in the end. Next person, Shawn He.
So good afternoon, everyone. My name is Shawn He. I'm a lecturer or assistant professor in computer science at the University of Birmingham. So my group, which is a small group, only consists of 8 PhD students, mainly focus on data mining, machine learning, and also optimization algorithms. And the most important thing is the complex network analysis. So basically, by collaborating with clinicians and biologists in Birmingham, we are developing algorithms to analyze omics data for biomarker discovery. We also develop algorithms to analyze biological networks, for example, protein interaction networks or co-expression networks and metabolic networks. So we have already applied our algorithms to glioma, which is a very nasty brain cancer. And we have already extract very informative information from these data sets. So if we are looking for opportunities to join consortiums to offer our techniques, computational techniques. So if you are interested, my name is on page 6 and line 1, 2, 3, 4, 5. Okay, thank you.
Thank you very much. Next person in line is Andre Jerez. No? Yes?
Yes.
Okay.
My name is Andre Jerez. I'm an employee of Syncom. Syncom is a contract research organization specialized in organic chemistry, coming from the northern part of the Netherlands, city of Groningen. We have a track record of over 25 years in assisting the pharmaceutical industry, mainly in early stage drug discovery programs. Currently we have about 100 FTE, a large portfolio of clients consisting of large pharmaceutical companies, biotech companies, but also recent startups from university and even academic groups. Main expertise of Syncom is synthetic chemistry, medicinal chemistry, process optimization. We offer integrated services, meaning that we also have offer ADME studies, specific in vitro and in vivo studies. Currently we are a partner in another IMI project, the European Lead Factory. So together with several academic groups and other SMEs, we are preparing a novel library for drug discovery. Main interest is to participate in drug discovery programs, but also in other programs where chemistry is involved. Thank you for your attention.
Thank you. Next person is Koka Yordahe. No? How about Desmond Johnston?
Thank you. This was written by my manager, so I'm going to read it in case I get it wrong. She frightens me. This is on behalf of Imperial College, which feels it could contribute to 5 of the key deliverables in this call. Firstly, Imperial is the National Coordinating Centre for a research network which recruits patients with type 1 diabetes within 6 months of diagnosis. So the research network, National Research Network, is one deliverable, and the sub-project recruiting people with type 1 diabetes, incident cases within 6 months of diagnosis, mostly within 6 weeks of diagnosis. We're currently recruiting 70 patients per month, and that will increase over the next 3 years to 100 patients per month. And all these people have given their permission to be contacted about research. The third deliverable, if I get this right, is 10 years experience of patient and public involvement as part of those network activities and other activities. The fourth one is that Imperial is the lead center for a national MRC, National Institute of Health Research Phenome Center, which is one of the largest metabolomic testing programs in the world. And then finally, Lab on a Chip. We have lots of expertise in that area we'd like to contribute. Thank you very much.
Thank you.
Next person is LaMotta.
So good afternoon.
Yes? Oh.
My name is LaMotta, and I'm a medicinal chemist, and I work at the Department of Pharmacy of the University of Pisa in Italy. That is an academic institution which is fully equipped to develop novel drug candidates through design, synthesis, and also evaluation till a preclinical level of novel chemical templates. Regarding diabetes, we focus on long-term diabetic complication affecting nervous, cardiovascular, ocular systems of diabetic patients, even when the glycemia is well controlled. And in particular, we target aldose reductase, whose inhibition may prevent the development of these complications. So we would like to share our expertise in this field to build up a multifunctional therapeutic approach to treat the diabetic patients. Thank you.
Thank you.
Next person is Sergio Larena. No. How about Les Mara? How about Gerd Martens?
Hi, my name is Heath Martins from the Diabetes Research Center here at Brussels Free University. I am a medical doctor and a beta cell biologist. My mission is the identification and clinical validation of novel biomarkers to detect beta cell destruction in the preclinical phase. Our research is basically very simple. Our center has access to primary human beta cells. On these we do proteomics to identify novel beta cell selective proteins, for which we then set up immunoassays and take these through serial validation in vitro, in animal models, and also in patients. For the work on patients, we can benefit from a very strong set of samples because our center coordinates the Belgian Diabetes Registry, which holds samples from preclinical high-risk patients, anti-CD3 treated patients at onset, and islet transplantation. At present, we can bring to the consortium 3 such markers. Also, we are studying proinsulin over C-peptide ratio as a biomarker for beta cell dysfunction in type 2 and pre-diabetes. So I stand here before you today because I ask help from the industrial partners to help me with immunoassay development, making monoclonal antibodies, and gaining access to high sensitivity immunoassay platforms on which my test can be multiplexed to be run ultimately on a single drop of blood. So thank you.
Thank you.
Thank you. Next person is Manuel Morilas. Okay, we go to the next. Krista Noehammer. Just a moment, the microphone is coming.
Hello everybody, my name is Kristin Oyama. I'm senior scientist at AIT, the Austrian Institute of Technology, which is Austria's largest research center. We are located in Vienna, which is, of course, as you know, a very nice place to have consortium meetings. AIT's molecular diagnostics unit, comprising of 50 people, I think has expertises most relevant to this upcoming diabetes call. which are as such, if you look at the specific objectives, we, for example, can offer a whole range of whole genome high-throughput omics technologies, which we can use to characterize prospective and retrospective type 1 patients. In addition, our special USP is that we can have a great expertise in genome-wide autoantibody profiling as well as DNA methylation studies. And we can also contribute bioinformatics and biostatistics expertise. And last but not least, we have a group which deals with development of diagnostic lab-on-chip devices. Thank you.
Thank you. Next, we have Mahalo Pope.
Hello, everybody. My name is Mahalo Pope, CSO of Terra Crystal, which is an innovative SME based in Romania. And we offer research expertise in the field of crystallization, polymorphism, and early formulation development. Our polymorph, salt, co-crystal, and pre-formulation studies include a number of unique elements. And I mentioned advanced crystallographic studies combined with molecular modeling, crystal engineering, and specialty analytics. We are fully equipped for that in order to enable very focused, customized screens and minimize, obviously, the risk of failure in further development of your new chemical entities. The message is that prior to applying very complex formulation technologies, we ensure that we discover the best possible crystal form in terms of stability, solubility, bioavailability, which ultimately will improve your in vitro and in vivo performance of the drugs. The role, actually, we are looking to be in an IMI consortium, and the role would be to help you develop effective drugs and secure and exploit the intellectual property via crystal engineering. Thank you very much. I'm sorry.
We have another person who perhaps signed up for the session as an accident, but I will still call his name, Mr. Bernd Stovasser.
Accident.
I think it was an accident.
Yes, I think it was an accident.
Yes, because he actually is someone from Sanofi and one of the speakers at the later session. So the next person is Kees van Bochove, or Boschove.
Yeah.
Hi, my name is Kees van Bochove. I'm the CEO of the Hive. I think this call needs a comprehensive, proven data warehouse for preclinical and clinical data, a translational research data warehouse which combines clinical and omics. I would say, don't reinvent the wheel, use proven open source technology. The Hive is an interdisciplinary team of computer scientists, bioinformaticians, data scientists that has stood up successfully implementation of open source data warehouses in companies like Sanofi, Janssen, Roche, but also working with IMI consortia such as EMIF and ETRIX. So what we can offer you is really that experience of how do you do that and how can you from day one start to actually load both external and internal data, prepare your omics pipelines with the experts in the consortia on that, and make sure that you spend your time on analyzing the data rather than doing nitty-gritty IT stuff, which has already been funded earlier by the European Union and pharma companies. So that's really my pitch, and I would invite you to maybe, if you're interested, to look at Transmart, which is one of the technologies we offer. It's a comprehensive open source technology for translational research. Thank you very much.
Thank you.
Next person is Scott Wagers.
Hello, I'm Scott Wagers. I'm the CEO of BioSci Consulting. And at BioSci Consulting, we believe that the big challenges in life sciences can only be solved by highly integrated research consortia functioning as virtual organizations. And this is an approach we developed in the first call project, UBioPred. For example, UBioPred has something like 43 partners, 300 some contacts that have been involved in it, and currently has 30 conference calls a month, which we facilitate. But more importantly, we also make sure that the important issues are addressed and solved and decisions are made and progress is made, which is one of the biggest issues you have to face in these projects. So our interest is in promoting that idea that projects should not be silos, but should be highly integrated. And so the way we're going to want to do that is be involved in more projects. So we'd like to be involved in this project in some way to help promote that vision. And we're also involved in the ETRICS, which Keys just mentioned, and we're involved in several other FP7 and MRC funded projects. Thank you.
Thank you.
We have two people left. One of them is Herman Rollens. No, not here with us. And the final person is Carmen Schwigeli.
Hello, everybody. My name is Carmen Schwigel, and I represent here a small privately owned company, Visera. Visera is a clinical research organization with excellent cooperation with researchers and clinical sites in Slovenia, Croatia, and Serbia. We are committed to be a first-choice clinical research organization offering on-time, high-quality delivery of clinical trials. We recognize the value of new treatment that addresses the patients with diabetes, and therefore Visera has already taken a number of actions to initiate the early clinical evaluation of new treatments in Southeastern Europe. And that's why we hope that IMI 2 initiative will result in conducting clinical studies also in this part of Europe. Thank you.
Thank you.
Thank you very much, everyone, for very informative elevator pitches, and thank you very much also for keeping to the time. We've been very efficient, and turns out we have some time left. So if any of you, perhaps any of you who haven't signed up for the elevator pitch would like to do it spontaneously, you have that opportunity right now. Just let us know. Raise your hand, please.
Thank you. My name is Pastor Endren from the University of Birmingham. I did put my name down for the elevator pitch, but clearly didn't make it onto the list. So I'm from Birmingham, which is in the Midlands in the UK. It's the second largest city and also very centrally based. I think there's two things I could offer as a partner. One is to do with cohorts. 7.5% of the UK population is South Asian, and Birmingham has a very high concentration of South Asians. They have the same incidence of type 1 diabetes as the background population. So half my transition clinic currently is South Asians, which reflects the area that the Children's Hospital is based in. South Asian type 1 is very poorly characterized, so we don't understand the genes, the antibodies, we don't understand the complication rates and some of the clinical demographics of this. And this is important because we know that in type 2 diabetes, South Asians have a more aggressive form of disease, so it's important that we characterize this in type 1. South Asians are typically difficult to recruit to clinical trials, and in Birmingham we have a track record of being able to recruit them to clinical trials. So we can provide that support. And that's all I want to say. Thank you.
Thank you.
We have another person, please.
Hello. My name is Radu Albulescu, I'm national representative on member group states, but now I speak for a group of research centered on the Institute of Metabolic Diseases and Diabetes in Bucharest, which can assist the project with experience in clinical study and also some additional expertise in proteomics that could provide help in the general area. Thank you.
Thank you very much. Do we have any other volunteers? Okay. Of course, yes. We also will take questions.
So, Heath Martins from Brussels Free University. I had a question on the general procedure. So, different consortia are stimulated to submit their proposal, and then this will be evaluated by the pharmaceutical companies.
No. Can I explain?
Oh.
Because this is really important. It will not be evaluated by the pharmaceutical companies. The expression of interest letters will be sent to the IMI, and the IMI Joint Undertaking Office has defined independent reviewers, minimum number of 3, which will review the expression of interest letters, and they decide. The FPR people have written the topic text, which you all have in your handouts. Up to now, it's out of us.
Okay.
It's very important.
Yeah, sorry. But what I miss, nevertheless, is that ultimately one consortium will be selected, so good groups will be included, but other good groups that might have complementary expertise will be not included.
Yes.
And so in some of these subdomains, there are excellent patient registries, excellent micro groups doing biomarker profiling or novel immune therapies. And so I think that the ultimate consortium that goes public would benefit from a second reflection round in which potentially missing elements in the winning proposal are identified, and that the procedure allows the opportunity to be expanded for key domains to people that were not selected in the first stage. I think that would be a wise decision.
Maybe I can take that question.
Maybe you take this question.
This was already discussed this morning. It is quite complicated to allow such process. However, in extreme cases, we've had examples where winning consortium was missing a key element, and the recommendation from the expert was to include certain expertise that was clearly missing. This is in rare cases. However, in this particular call, I don't know if you have read the topic text, there is an opportunity for a restricted call at a certain point, given that the first phase of the project goes according to plan and there is a possibility to expand the project and indeed do some clinical work, then there will be expansion of this project with new funding coming both from the companies and from IMI, and perhaps opening the consortium for additional partners for the expertise which is lacking.
And will the independent reviewers see all proposals?
Yes.
So that they will be able to fully compare?
Yes, of course,
yeah.
of course, yes.
All right.
There is another question here, and then in the back.
Thank you. Just to follow up on that question, are the evaluators going to be taken from the Horizon 2020 pool of evaluators, or is there going to be a separate call for that?
Normally, the way we try to identify experts, we of course look at the database of the Commission, but not always it is possible to find suitable experts, especially in the field which is such like diabetes, where people are collaborating quite closely. So it is difficult to find people who are truly independent. So we try to reach out sometimes to other countries, US, and we try to find experts which are really leaders in the field. Also, to keep in mind, since this topic is quite broad and have multiple aspects, we try to also cover those different aspects. So perhaps there will be someone who is expert in diabetes, in immuno-inflammatory part. There might be someone who is a regulator. There might be someone who is expert in data management and so on. It's not a call. This is normally done by us, by the scientific officers. We look at literature, we look at experts in the field and try to identify experts. We invite them personally. There was a question in the back.
Kees van Bokhoven from the Hive. I just gave my pitch, so maybe now you have the background for my question. I noticed that JDRF also has an open source platform for storing T1DM data, which brings me to the question from FPR view, is there any data like existing cohorts that you would bring into this consortium that could be leveraged from the start?
I would answer this with yes.
Okay, but do you have any specifics on that? Because I think it's quite relevant, you know, for the database point of view, and also to see what we can build on. I'm sure that FPR companies have quite some trials to offer. So
I think there are a lot of academic people who have a lot to offer because they have already big cohorts and have collected biological samples for a long period of time already. So this information should be right away available, as well as from the pharmaceutical side. If we have data on our already performed trials we should learn from, that then they will be also a part of it. In regard to your earlier point where you said, why should we something develop new if it has been already developed in regard to data analysis? I think this is also our clear understanding. Nobody needs to invent something if it's already there. It is very important to have a database which allows very complex correlational analysis, but we also should learn from what has been already established within the IMI calls, where also databases have been created for the different topics. So we should look also what is already available instead of starting from stretch zero again.
Yeah, thank you.
Do we have any other questions? Here, please.
The microphone is coming from the back.
So you mentioned, thank you, in your introductory slides on the cohorts you were looking for. We've discussed some of those. But I wondered whether, you know, you saw that there was an opportunity to go a little bit further maybe in some of those cohorts. Some might be controls, which are often difficult to match perfectly. But in particular, I referred earlier on to the question of risk. And we still don't know, for example, exactly how much beta cell function protects you from what complications, from ketoacidosis risk. So it becomes a little bit more difficult to say what is worth saving, what is not worth saving. And as we're trying to decide on more aggressive interventions, it becomes quite important to know what we have to save, if that makes any sense. So in terms of trying to decide people at the most metabolic risk, this is phenotyping children and adults with diabetes for their long-term risk, if you like, with different levels of beta cell function, rather than just looking at their immune profile.
Right, so we do need something to have a better readout over the beta cell status within those at-risk patients. Absolutely. So the sample collection should be everything what you can collect from a child, from a person who's alive. So it will be urine, it will be plasma, it will be serum. And then you have to see what kind of molecular techniques you will have to apply to get the most information on the status of the beta cell. And I know it's very exploratory at this point. Does this answer your question? No, I don't think so. Not quite. Okay. I'm sorry.
For example, no, it's very interesting. For example, the risk of ketoacidosis will increase the less insulin you have.
Yeah.
And defining the people, the kids and adults at the highest risk of ketoacidosis may determine people in whom you might want to say protection of preservation of that amount of beta cell function will be valuable. So I'm thinking about their diabetes risk rather than so much about their immunological profile.
Okay. So I cannot answer the question. I don't know what would be the characteristics to get those patients and how likely this will be to include those patients.
I guess what I'm thinking about is if you were to introduce an intervention, and you said that some of the companies do have interventions, to decide on who you would treat, you need to decide whether they would respond, certainly, but you need to decide what is the risk for that individual child.
But we also think that when we pull 4 years of research in this area to better understand the early patients, then we will better understand what would be the best person to select for the asset we are planning to apply.
Okay.
That's why we do need to work for 4 years before we can decide this.
Thank you.
Are there any other questions? Okay, so I invite you to use this opportunity and network. We will have a 40 minutes coffee break, just to inform you that the next session in this room starts at 4, not 4:30. Apparently both of our workshops have been very efficient on time, so we are moving everything up by 30 minutes. So if you are interested to hear about future calls of IMI2, there will be a session here in this room at 4. Have a nice coffee break.
Thank you very much.
Thank you.
Children